Welcome to science class. Today we are going to be looking inside of the eukaryotic cell and describing the many functions that its organelles play. Organelles are wonderful things, and there are many of them. The name means little organs, and that's a good comparison. No single organelle can sustain all of the activities necessary to keep a cell alive, and neither can any of your organs. Organelles come in a wide variety of shapes and structures. Many are composed of lipid membranes, but several are made of protein structures. Ribosomes are even made of nucleic acids. There are maybe 30 or more of these structures that can be called organelles, and we won't be covering all of them today. I want to cover the ones whose functions will be most important and relevant going forward in this class. Remember, life is cellular, and all the activities that a living thing does are carried out at the cellular level. These little organelles are what do the individual jobs of cells. With all that being said, let's get started. We will be discussing a lot of organelles today. A few major ones will get special attention, and about the same number of minor ones I'll try to be brief with. Now, not every cell is the same. Prokaryotes only contain ribosomes as far as organelles go. Our focus is on eukaryotes today, but there are several types of eukaryotes. We have 4 kingdoms of eukaryotic life, therefore 4 different kinds of eukaryotic cells: the protists, plants, fungi, and animals. Protists are single-celled organisms. Some are heterotrophic, some are autotrophic, or in other words, some of them eat and some of them photosynthesize. Plants are multicellular autotrophic organisms. Fungi are heterotrophs and can be unicellular or multicellular. Fungi are more closely related to animals than they are to plants, even though you can buy them in the produce aisle. And animals are multicellular eukaryotes with an internal digestive system. Fungi have an external digestive system. Most of the major organelles I will cover today are present in all eukaryotic cells, whereas some are exclusive to just 1 or 2 kingdoms. We're going to start with energy production. Plants are autotrophs. Now, autotroph doesn't really mean they make their own energy because plants don't make energy. They make food, and they use their cells to metabolize that food and extract energy from it. The organelle that allows plants to make their own food is the chloroplast. The chloroplast is usually a green organelle, and it has a special pigment called chlorophyll within it that absorbs light, borrowing the light energy to break apart water molecules, synthesizing carbohydrates from carbon dioxide molecules. Oxygen gas is lost as a waste product. All plants contain chloroplasts and many protists. In fact, protists like diatoms and dinoflagellate are some of the most numerous organisms on the planet, and along with other phytoplankton, create most of the world's oxygen gas. Plankton is a term used to describe any kind of organism that is immobile or drifts, is unicellular, and photosynthesizes. Some bacteria fall under that category as well. Those carbohydrates made by the chloroplast are metabolized and used to make energy in the form of ATP by mitochondria. Virtually all eukaryotes, apart from some unicellular ones, have mitochondria. Almost the exact opposite of what happens in the chloroplast happens in the mitochondria. Carbohydrates are broken apart with the aid of oxygen gas to create large quantities of energy in the form of ATP, a molecule that all living cells use to do every kind of cellular activity that requires energy. Carbon dioxide and water are produced as waste products in this process. We will get into much greater detail about the processes of the mitochondria and the chloroplast in another video. Amazingly, the chloroplast and mitochondria have their own DNA and reproduce on their own. Your cells cannot make new mitochondria. They undergo their own binary fission. We are fairly sure that mitochondria and chloroplast are bacteria, or they were and are something else today. It's hard to call a mitochondrion a cell, or a chloroplast for that matter, but in many senses they are. This is an idea called endosymbiosis, and I promise we will talk about it more when we cover evolution. Next up, the nucleus. The nucleus distinguishes eukaryotes from prokaryotes. It is a double membrane structure that houses the DNA of eukaryotic cells. The nucleus is covered in pores, which allow RNA to exit the nucleus, along with ribosomes. RNA is a single-stranded nucleic acid molecule built from our DNA template. Having a nucleus protects the DNA from interference. Prokaryotes can multiply at an unbelievable rate. A single bacteria can give rise to 140 trillion descendants in a 24-hour period under perfect conditions. With such a prolific rate of replication, errors are tolerated. There will always be survivors to pick up the slack. Eukaryotes are more complex and require more control over their division, and the nucleus ensures that the DNA is better protected from deleterious or otherwise harmful mutations. Inside of the nucleus is a protein structure called the nucleolus. This structure makes the ribosomes I mentioned a bit ago. The nucleus, DNA, ribosomes, and another organelle all tie in together rather closely to accomplish a specific goal, which I'll explain in a little bit. Attached to the nucleus is the endoplasmic reticulum. This is a network of folded lipid membranes called cisternae that serve several functions. You find ribosomes attached to much of the ER. This creates what's called the rough ER. This is the site of protein synthesis. You may remember that proteins are the most abundant organic molecule in your body, and they have by far the greatest diversity of function. Proteins are the functional molecule in our bodies. They can be enzymes, they can be structures, they can be hormones, they can be neurotransmitters, you name it. Many ribosomes freely float in the cytosol or fluid regions of the cell not attached to anything. For a ribosome to build a protein, it reads an RNA strand. That RNA strand again comes from the DNA, stored in the nucleus. The rough ER not only serves as an organized site for protein synthesis, but also protein transport. The lipid membranes that our organelles and cell membrane are made of are highly flexible, and they can change shape and fuse with other lipid membranes. Kind of like this.
Barnacles, what could be worse than a giant paint bubble?
Oh, I know. 2 giant paint bubbles.
No!
When a protein is built on the surface of the rough ER, the ER membrane will pinch itself off, creating a vesicle for the protein to be transported in. There is endoplasmic reticulum that does not have ribosomes attached to it, and we call that the smooth ER. It's structurally different as well. It forms more of a tube network as opposed to a close-knit series of flattened stacks like the rough ER. The smooth ER builds lipid compounds and detoxifies substances for our bodies. Building lipid compounds is an extremely important job. Lipids are the second most abundant organic molecules in our bodies, forming our cellular and organelle membranes. But cholesterol and steroids are lipid molecules as well, and they serve as the basis for the majority of hormones. We need to pause here to point something out. Look at these electron microscope images of the ER region of the cell. Notice how incredibly dense with material this is. There are no wasted spaces. Cell models in books do not accurately depict cell structure. They just make organelles easy to identify. Chemistry can only occur if there is a surface for the reaction to take place. Our cells waste absolutely no space because they are undergoing hundreds of millions of chemical reactions per second. This organization is crucial for understanding the activity within cells and the level of activity that goes on, because it's mind-boggling, but the standard method for learning these things typically does not do that justice. The next organelle is the Golgi body. The Golgi is similar to the ER in appearance, but the Golgi is much more of a shapeshifter. Its cisternae are more highly distinct as well. The Golgi receives vesicles. Those vesicles may come from the ER, or they may come from the cell membrane, pinching inward and accepting material from outside of the cell. The Golgi modifies material that it receives, which are typically proteins. The molecule will be altered to finalize its shape and function, partially broken down, or a number of other modifications. The Golgi sets the final destination as well for the material. It's a bit like an assembly line, a quality check, and a post office all in one. I've been talking about how cells transport material around within them. How does that work? Again, we picture the cell as filled with fluid and all these organelles float inside of it. What gets the vesicle from the ER to the Golgi? Blind luck? No, and again, our models are inaccurate. There are a host of different protein filaments that crisscross throughout the entire cell. These protein filaments are the cytoskeleton of the cell. The cytoskeleton has a host of functions, but I will cover just a few of them. In a beautiful and bizarre way, some of these filaments serve as a sort of highway network in our cells. Vesicles are transported by a type of motor protein called kinesin. These proteins grab onto the vesicle and then, in a literal sense, walk along the cytoskeleton filament to their destination. This is how vesicles get to where they need to go. Larger cytoskeleton filaments support the organelles, preventing them from tangling with each other and just holding them in place in general. Other kinds of cytoskeleton are involved in movement. Muscle contraction involves myosin and actin proteins latching onto one another and sliding together, almost like pulling on a rope. Flagella, the whip-like tails of spermatozoa cells, bacteria, and other cells, have a particular cytoskeleton arrangement within them. Cytoskeleton filaments are also used in cellular division, which we will cover in detail in yet another future video. Animal cells and a few plant cells contain lysosomes. Lysosomes are used to break down material and put it into usable form, to kill bacteria and recycle old organelles. Remember, 96% of your body is made of just 4 atoms. By breaking down biomolecules, we can repurpose the building blocks to recreate virtually any part of you. The last organelle that I want to cover is the vacuole. Plants will tend to have one large vacuole. This vacuole holds water, salts, electrolytes, and other compounds, but mainly water, and it provides the rigidity of the plant. When plants wilt, it is because they are losing water, like an air mattress that has lost air and now sags. If you've ever taken celery or broccoli out of the fridge and noticed that it is droopy, the food probably isn't spoiled, it just needs some water. Place it in a bowl of water and it will swell right back up and be crunchy again. Animals and protists have vacuoles, but they don't quite serve the same purposes. Single-celled eukaryotes will contain a contractile vacuole. This is a pump that fills with nothing but pure water and then pumps it back out. Otherwise, the cell would fill up with water so much that it would burst. The reason that happens is the subject of a video coming up shortly. The big picture here is that cellular life is very complex. As complex as you are as a living organism, your individual cells rival you. No, they are not conscious, but the way they can take in the random matter of the universe and create the chemistry of life out of it is equally as beautiful and mesmerizing as anything I can think of that a human brain has produced. All of these organelles have a specific function. You can't really say that they have goals or that they're following a plan, it's just chemistry. But they produce you. You! A living, breathing, intelligent, awesome person. If you ever get a chance to observe some of your cells under a microscope, I hope you can't look at them in any kind of ordinary sense, because they are anything but ordinary. Next time, we are going to be taking a look at the membranes that form the boundaries of our cells and discuss how things get in and out. Thanks for watching.